All right, ladies and gentlemen, we need to continue our discussion of the central dogma of biology and pick up where we left off, where we talked about how DNA, which is the information that contains everything that's needed to create you, is copied into RNA and then eventually ends up as proteins, which lead directly to your traits. The thing is, this whole step in the middle about RNA is a bit more complicated than just DNA as a complementary copy made. So what we talked about in our last video is that DNA is transcribed, and transcription creates messenger RNA in the nucleus. And messenger RNA is a complementary copy of DNA. Okay, so that's what happens during transcription. Well, after transcription is over, this mRNA leaves the nucleus and it ends up with ribosomes. Now, hopefully you remember that ribosomes are the organelle responsible for protein synthesis. And so what we're dealing with here is that there's a connection between DNA's information and it leads us to proteins. Well, what's going on here is this step in the middle of RNA is how the information of DNA is actually going to lead to protein synthesis. Okay? Now what happens is that the ribosomes, the messenger RNA interacts with a different type of RNA that's known as tRNA. And tRNA stands for transfer RNA. And we're going to talk more about that later in this presentation. Now, what happens here is the interaction between messenger RNA, ribosomes, and transfer RNA provides proteins with their structure. And as we know, because we've said this over and over again, structure equals function. So if this provides proteins their structure, it also provides proteins their function. And this is how we get all these different proteins that do all of these different jobs. So let's talk a little bit more about the details as to how this works. So we know that transcription creates a complementary copy of DNA, and that complementary copy is in the form of something called messenger RNA. So you can see right here, what's written in blue is DNA. You have the strand ATC, GGG, CGC, TTA, ATA. And then underneath what you have is a complementary messenger RNA copy, UAG, CCC, GCG, AAU, UAU. Remember that you have the U because there is uracil in RNA but not in DNA. Now, the interesting thing about this is that messenger RNA is a copy of the DNA and it contains the information for our traits. But since this is written in a language, we need to know how to read it. And what's cool is that it's written in groups of 3. And each group of 3 is called a codon. So what that means is that when I want to read this, I need to look at it like this. And each group of 3 provides me what's called an amino acid. An amino acid are the pieces of proteins. And there's 20 of them. Okay? So how do I read these groups of 3? So here we have again, proteins are made of units called amino acid. Now each one of these codons gives me the information for one amino acid. And so we have this thing called a codon chart. This actually comes in 2 forms. You can see it like this, in this circle shape, or you can see it in, sometimes there's a box where you would go up one side, across the top, and down the other. But what we have here is, this is the DNA. Okay, so this right here is the DNA, and this is the mRNA complementary that comes from it. You know how I can tell that? T for DNA, U for RNA. So what happens here is I look for AUG. A, U, G. You start in the middle, go to the second level, go to the third. So this codes for the amino acid methionine. And I find CCC, C, C, C. And that gives me proline. Now, what you can see right here is CCC gives me proline. CCA, CCG, CCU all give me proline. So you can see by looking at this chart that there are many examples where more than one amino acid, or rather, I'm sorry, more than one codon codes for amino acids. So here we have GGG, so GGG, that gives me glycine. UGC, UGC, gives me cysteine. GGC, so here you go, here's a great example of one of those where two different codons can give me the same amino acid. And then UAG. Now you notice here that we have stop, and it's also here, okay? This is how the ribosomes know when to end the process of creating proteins, is when they hit one of these codons that says stop. There's actually 3 stop codons, and when the machinery of the ribosome hits one of these, then the ribosome separates from the messenger RNA and actually just stops making the protein, and then everything before it becomes the amino acid sequence. This right here, methionine, is actually the start codon. So every single amino acid begins with methionine. And every single protein begins with the amino acid methionine, and every single protein ends with one of these 3 stop codons that are right here. All right, so let's talk a little bit about how these codons are read. So proteins are assembled at the ribosome. That's something we learned way back when we were discussing the organelles of the cell. But what we're actually saying is that messenger RNA, the codons of it, are read at the ribosome and turned into amino acid sequences. Okay. So what we're going to do is we're going to say the ribosome creates amino acid sequences. Okay, that's what the ribosome does. Ribosomes have 2 main parts. There's what's called the large subunit, and we call it that because it's the bigger of the 2 parts. And there is the small subunit. And what you'll notice is that ribosomes actually the 2 subunits sort of sandwich in between them the messenger RNA. So this blue strip here is messenger RNA, and the 2 pieces just sort of like push against each other and keep the messenger RNA in the middle. Ribosomes are actually made of 2 different key parts. First off, they themselves are made of proteins, and they are also made of something called rRNA. So this is the 3rd and final RNA type, and it stands for ribosomal RNA. All right, so there's 3 types of RNA: messenger RNA, transfer RNA, rRNA, or ribosomal RNA. Okay, so there's the basics of the ribosome. Now, this is our friend tRNA, or transfer RNA. And transfer RNA is single-stranded. You can see right here the 3' and right here the 5'. So it actually is just one strand that runs all going to skip that for a second. All along here. All right. Now, what happens with transfer RNA is that up here, there is one amino acid attached. Okay. Down here, we have what's called an anti-codon. All right. Now, the one amino acid attached depends on the anticodon. All right. So which of the 20 is determined by the anticodon? Now, the anticodon is actually complementary to one of the codons. So, for instance, if I had a DNA sequence that was that was ATC, the messenger RNA would be UAG, and this is what we find on the codon chart. Well, the tRNA over here at the anticodon site of this would actually have a UG. And then at the top here, that would give me a specific amino acid. And then which amino acid just depends on which one this codon gives me the information for. And that's the kind of thing I would look for on this chart. So if I'm looking at a UAG, that's the stop, so there actually wouldn't be an amino acid up there. So right here, this is actually how the whole thing comes together. So what happens is this, and you can see it right here, is my messenger RNA strand. This is the small subunit, large subunit. This is what we call the A site. This is what we call the P site. tRNA enters at the P site and leaves at the A site. What happens is this piece is no longer attached to this amino acid and leaves. This piece comes in and brings in an amino acid, and the ribosome attaches the amino acid to each other. And what's key is it does this in a specific order determined by the mRNA. And that order gives proteins their function. All right, I guess that's the basics of translation.